All right, remember that a monohybrid cross is only a cross between individuals that involve one pair, okay, of contrasting stripes called monohybrid cross. So mono meaning one. We know that, all right? The simplest way to predict the results of monohybrid cross by using a Punnett square was named after the inventor Mr. Punnett, okay? Here is an example of a Punnett square. It has 1, 2, 3, 4. 4 squares. Each square is 25%, okay? In this case, equaling 100%, okay? Moving on. An example between a monohybrid cross is a cross between true breeding plants. So remember that they gave us, if it's tall, it's dominant, okay? If it's short, it's recessive, all right? In this case, the letters that we're going to be using representing these two plants are going to be uppercase if it's dominant, lowercase if it's recessive, all right? So in this case, a cross between a true breeding plant for tallness with a true breeding shortness. So tallness, true breeding, meaning it's going to be 2 uppercase T. True breeding shortness would be 2 lowercase t. So in this case, we would be performing that Punnett square. Okay, so let's move on to our second one. A cross between 2 heterozygous individuals. Remember that heterozygous is different. So in this case, we would have a uppercase T, lowercase t times an uppercase T, lowercase 2, because it says 2 individuals are heterozygous. All right. Third example, a cross between a homozygous dominant. Homozygous, you already know homozygous is different. I mean, it's the same, all right? So homozygous dominant would be 2 tall T's, because remember, the big T's are dominant, times a heterozygous parent, uppercase and lowercase. Anytime that it says heterozygous, it's always going to have an uppercase letter and a lowercase letter, all right? Depending on what letters the example is using. So let's try this first example. Let me change my color. All right, a cross between a true breeding plant for tallness. Okay, so in this case, we're already thinking of our letters. So it's going to be 2 uppercase T times a plant that is true breeding shortness. Okay, so in this case, it would be 2 lowercase t. So let's put our letters in our Punnett square. It doesn't matter where you put your letters, honestly, as long as you're combining them correctly. How do you combine them? You're going to take this first letter times this letter. So you're going to get T and T. All right. We're going to combine this letter with this letter. We get uppercase and lowercase. All right. We're going to combine this letter with this letter. We get uppercase and lowercase. And then we do that same thing with this one. We're going to take this T with this T and we get uppercase and lowercase. Okay. Always make sure that you have a, you put the uppercase, which is our dominant first, okay? And not the recessive, and always combine them together. All the letters that are the same go with the same letter. So these are the same alleles, okay? So that is the first example, but now we have to look for the genotype ratio and the phenotype ratio. How do we get that? The genotype ratio is basically how many different letter combinations do you have in each square. So let's take an example. This first square has an uppercase and a lowercase t. Out of these boxes, how many squares do I have that look exactly like that? So we have 1, 2, 3, 4. Okay, so in this case, we have 4 out of 4 equals 1 out of 1. Reduce it. All right. Phenotype ratio is, in this case, we're comparing tall plants versus short. Okay. All right. So if it's a tall plant, it's going to have a dominant T as the first one. Okay. If it's a short plant, it's going to have a lowercase t as the first one. All right. So in this case, let's look at these 4 examples. So out of these, how many are going to be tall plants? So we have 1, 2, 3, 4. Okay. Out of these, how many are going to be short? None, okay? Because we don't have any that have a lowercase as the first one. Alrighty? That is how you get your genotype and phenotype ratio. Let's move on to our second example. A cross between two heterozygous. You already know heterozygous is different, okay? In this case, we would have an uppercase T, lowercase, times uppercase, lowercase, okay? Because it's heterozygous. So let's combine our letters. Alright, let's perform it. You can check just in case you already did it. Alright, perfect. Okay. So now let's get our genotype ratio, phenotype ratio. Remember again that genotype ratio is the different combination of alleles or letters that you have in each box. So let's look at this first one. Out of this first box, how many look like that? 1. Okay. Let's move on to our next example of allele letters. Out of these 2 letters, how many look exactly like that? We have 2. Okay. Moving on to our next one. Out of this combo right here, how many boxes do we see that look exactly like that? Only 1. Okay. That's how we get our genotype ratio. Phenotype ratio is, remember, we're comparing tall versus short. Okay. Out of these, how many are going to have a recessive trait? Okay. How many of these are going to be tall? So we have 1, 2, 3. Okay. We have 3. All right. Out of these, how many are going to be short? If it's short, it's going to have a recessive trait, and we only have 1. Okay. How are you going to know if your genotype and genotypes are correct? If they're equal 4. If they equal 4, it's meaning that you have the correct numbers, okay? Because we have 1 box, 2 boxes, 3, and 4. All right. Moving on to our next example right here. A cross between homozygous dominant, okay? Homozygous, we already know homozygous is same. Dominant in this case, it would be uppercase, okay? Uppercase T. And a heterozygous individual. We already know heterozygous is different, okay? In this case, we're going to have an uppercase T and a lowercase t. So our letter combo would be 2 homozygous dominant times 2 times a heterozygous trait. So let's put our letters. All right. Let's perform it. Let's get our genotype ratio, phenotype ratio. Again, let's look at the letter combos. Out of this letter combo, how many do we have that look exactly like that? There are 2 homozygous dominant. In this case, we have 2. Okay. Moving on to our next allele combo, or letters. Out of this letter combo, how many do we have that look exactly like that? We have 2. Okay. Phenotype ratio, you already know we're comparing tall versus short, okay, plants. And let's put our 2 letters that represent that. Uppercase for tall, lowercase for short. Out of these 4 combos, how many do we have that are going to be tall dominant plants? So let's check it out. Tall dominant means if it has a uppercase as the first one. Okay, in this case, we would have 4 tall. Okay. Out of these 4 letter combos, how many are going to be short? If it's short, it's going to have a lowercase t as the first one. In this case, we have none. Okay. 0. Does it equal 4? Yes. Does it equal 4? Yes. All right. That is how you perform a monohybrid Punnett square. Alrighty. Well, now we're going to move on to dihybrid Punnett squares.